What if I told you Pac-Man could be the answer to treating COVID-19? Hard to believe, right? Of course, I'm not talking about Pac-Man the game, but instead Prophylactic Antiviral CRISPR in human cells, or what scientists call Pac-Man for short. It's a mouthful, let's break it down. First off, prophylactic is a course of action to prevent disease. Just as you would use sunscreen to prevent a sunburn, you would use a prophylactic treatment to prevent disease. Furthermore, antiviral is a type of drug or treatment that is effective against viruses. But what exactly is CRISPR? CRISPR is an abbreviation for clustered, regularly interspaced, short palindromic repeats. Once again, let's break it down. CRISPR is a special sequence of DNA found inside bacteria. In each CRISPR sequence, there are segments of DNA that repeat itself. Separating each repeating sequence is a spacer DNA, which is always a unique sequence. This spacer DNA is extremely important because it matches the DNA of viruses, allowing CRISPR to recognize the virus. In some cases, scientists are actually able to genetically modify CRISPR by changing its spacer DNA to match that of viruses, including influenza, and eradicate them in laboratory environments. CRISPR also uses Cas genes that code for Cas proteins. Together they make a CRISPR-Cas complex. Depending on the sequence of Cas genes, a certain protein will be made. One type of protein, known as a Cas9 protein, is responsible for destroying the virus by snipping the virus's DNA. However, COVID-19 is slightly different. Instead of affecting human cells by using viral DNA, it uses RNA, which is essentially one half of a DNA strand. This is why the type of Cas protein is extremely important. Since Cas9 targets viral DNA, it can't destroy COVID-19's RNA. However, a new protein, Cas13 protein, targets viral RNA sequences, allowing it to destroy COVID-19's RNA. Without its RNA, the virus is unable to hack into the cell and create more copies of itself, allowing patients with COVID-19 to recover more effectively. And now we arrive at the biggest problem of all. How do we deliver this CRISPR-Cas13 system to the human cell? One method was through utilizing lipidoids. Lipidoids are tiny containers that hold the CRISPR-Cas13 system and deliver them to the human cell. Think of lipidoids as a military airplane. The lipitoid airplane is what actually gets the CRISPR-Cas13 soldiers to the battlefield or where the infected cell is. The RNA acts like a tracker and finds the exact location of the enemy virus on the battlefield or infected cell. The CRISPR soldiers are essential to fighting the enemy virus and must be a ranking of Cas13 to destroy them. When lipidoids were put to the test, Stanford researchers found that they successfully delivered Pac-Man to human cells and had reduced synthetic COVID-19 molecules by more than 90%. While Pac-Man provides promising results, a lot more testing is necessary to ensure its effectiveness and to provide a delivery method. But no need to worry, right now there are scientists hard at work on Pac-Man.